Okay. Some people did not come to lab this week, which in this week's lab consisted of basically the pre-lab for next week, right? So in this week, I discussed how to do the lab, and I also discussed how to do a lab report. So if you didn't make it this week, then you got a 0. But even worse, that means you also can't come next week, which means you have a 0 for next week. And even worse than that, then you'll have to write a lab report that's also worth 20 points. And if you're writing a lab report on a lab you didn't do, that's going to be even harder. You have to get data from your classmates, so on and so forth. So here's what I'm going to do. I'm going to do this lecture yet a third time. I'm going to record it. This way the people that missed the lecture, they can watch it now. They can prove they watch it by answering some questions. And then they can get partial credit for this week's lab, because I'm not going to give you full credit if you didn't come to lab, but I will at least give you partial credit. And then next week you can come to lab. So again, to be clear, of course, if you're watching this, I'm probably preaching to the choir, but if you do not watch this video and prove that you watched it, with answering the questions or whatever, then you will not be able to come to lab next week and you will get another 0. So just keep that in mind. Anyway, let me get started here. I just downloaded the PowerPoint. Let me pull it up. Well, I thought I did. Let's see, downloads. There we go. Let's take a look. Also, I noticed there was some mistakes with the last version that I was sharing during lab itself. So I'll be able to edit that while we're doing this one. For example, let's get rid of that date. That is no longer relevant. So here we go. Let's take a look here. This is all about the photosynthesis respiration lab. And again, like I said, if you weren't there this week, in theory, you get a 0. But I will allow you to watch this video and answer the questions and at least get partial credit. And again, you have to watch this and answer the questions in addition to doing the pre-lab before you can come to lab next week. So if you don't do all that stuff, you get a 0. Let me also change this. This is another thing that was outdated. Excuse me. Today's lab is based on attendance, right? So full 20 points for being on time and leaving at the end. Of course, that isn't relevant if you're watching the video because you're not on time and you're not leaving. You're just watching the video. And of course, you can't be late or leave early because, again, you're watching the video. So there we go. So next week will be just the way it normally works, right? So next week you have a pre-lab to do that's due on Sunday that's worth 5 points, and it also includes 2 videos. You cannot come to lab unless you do the pre-lab, including the videos, in addition to this video, right? So you have a lot on your plate. So yeah, anyway, 5 points for the pre-lab as usual, 15 points for the lab worksheet as usual. And then afterwards, you will have a 20-point lab report that you need to write. And I'm going to teach you how to write that in this video. So here are the 4 questions that we're going to answer in this lab. The first one we're going to answer is, do photosynthetic organisms photosynthesize faster in more intense light? To simplify that, for example, if we were talking about plants, you could say, what plant is going to photosynthesize more? A plant that's really close to the light, or a plant that's really far from the light, or a plant that's somewhere in between? Think along those lines. The next question we're going to answer in this lab is, do photosynthetic organisms respire when light is absent? So this is a respiration question, not a photosynthesis question, right? So here we're going to see if they respire when there's no light. Now, in theory, we should expect that to happen, right? Because respiration, as you know from lecture, only requires oxygen and glucose, basically, right? Oxygen and glucose, and you're good to go. You don't need sunlight for respiration, right? We all respire as humans, but we don't require sunlight for that purpose. So anyway, that's just something to keep in mind. The third experiment, the question that we're going to answer in the third experiment is, do photosynthetic organisms photosynthesize and respire faster at warmer temperatures? In other words, does temperature affect the rate of photosynthesis and respiration, right? So in a sense, these are two different experiments you're going to do wrapped up into one. You're going to see how temperature affects photosynthesis, right? You're going to see how temperature affects respiration. And then the last question, that we're going to answer with the fourth experiment is, do photosynthetic organisms photosynthesize at different rates in different and lights of different wavelength? Now, wavelength basically means color, right? Different colors have different wavelengths. So another way of asking that question is, does the color of light affect photosynthesis? Right? So does things photosynthesize better in green light or white light or red light or blue light? Right? That's the last question that we're going to answer in the fourth experiment. So then we say, well, how do we answer these questions? Right? How we have these questions, how do we answer them? So if we're talking about it from the perspective of the scientific method, we skipped the first step, which is observation, right? We've gone directly to the question, right? And next would come the hypothesis, but we'll discuss that later. Right now, let's talk about how can we answer these questions, right? Now, if we had all semester, we could grow some plants. For example, the one about the different colors of light, right? We could get 4 plants starting from seed. We grow one in a white light for, 10 weeks. We'll grow one under blue light for 10 weeks and grow one under green light for 10 weeks, so on and so forth. But we can't do that, right? We don't have 10 weeks. We have 1 hour and 50 minutes. So how can we answer these questions? And before we can talk about that, let's talk about a little background information that you should already know at this point, at least for respiration. But we'll probably talk about we will talk about photosynthesis first. But here's the basic equation for photosynthesis. When you mix water and carbon dioxide, you get glucose and oxygen, right? So if we had the means to do this, well, I'll go ahead and tell you this little spoiler alert. We'll talk about this later. But you're going to have these little tubes in them with liquid and little algae beads in that liquid, right? That's what we're going to use to measure this, right? So let me go ahead and again, that's just a little hint of what we're getting at. But this is liquid, right? And this is all just a bunch of liquid that those things are in. So if we could somehow measure the amount of water in there, then if the amount of water was decreasing, we would know photosynthesis is happening, right? Because photosynthesis uses water. If we could measure the amount of carbon dioxide in that liquid, and if it was going down, we would know photosynthesis is happening because it's using carbon dioxide. If we could somehow measure the amount of glucose in those algae beads or perhaps in the liquid, and if the glucose levels were going up, we would know photosynthesis is happening because photosynthesis makes glucose. Likewise, finally, if we could somehow measure the oxygen content in the liquid or even in those little algae beads, and the oxygen rate was increasing, then we would know photosynthesis is happening, is because photosynthesis increases or produces oxygen, right? And the opposite is true of respiration. If we could take measurements, we found out that glucose levels were dropping. If glucose levels were dropping, then we know respiration is happening, right? Because respiration uses glucose. So if oxygen levels are dropping, must be respiration happening because respiration uses oxygen. If we could measure carbon dioxide levels, if the carbon dioxide levels were increasing, we would say respiration is happening because it produces carbon dioxide. And then finally, again, if we could measure the water level, the water content, we would find the water levels or content would be increasing because respiration produces water, right? So these are some basics, right? So the question is then, obviously I'm going somewhere with this. So what are we going to be measuring, right? We're going to be measuring glucose, oxygen, carbon dioxide, or water. Any one of these things, if we could somehow measure them, then we know, right? Again, just using this as an example, because right here, we know if water increases, it's respiration, and if water decreases, it's photosynthesis, right? So which one are we going to use? Well, I'll give you a little hint here. As you know from our previous discussions, when you mix carbon dioxide and water, it produces an acid. Yes, technically it's carbonic acid. I don't really care that you know what kind of acid it is. It doesn't matter. But when you when you mix carbon dioxide and water, you produce an acid. Therefore, photosynthesis, as you know from here, photosynthesis reduces the levels of carbon dioxide, right? Because photosynthesis uses carbon dioxide. And if it's reducing the levels of carbon dioxide, that means it's going to reduce the amount of acid in the solution, right? And what happens to the pH when you reduce the amount of acid, right? When you make something more basic and less acidic, what happens to the pH? The pH increases. Let me fix this. Is making a noise. Let me check on this really quickly. Sorry for this. I'm gonna pull this up and X out of there. There we go. Yeah, I had to fix that. Anyway, getting back to this. Actually, let me fix this. No, that's good. Yeah, here we are from current side. Sorry about the distraction there. Anyway, like we said, photosynthesis uses carbon dioxide. Carbon dioxide, all right, so therefore we're reducing the amount of carbon dioxide in the solution. Therefore, we're going to be reducing the amount of carbonic oxide in the solution. Therefore, the pH is going to go up, right? So when we are doing these experiments, and I'm going to explain how we're going to do the pH, but for now, just know that when we're doing these experiments, if we find the pH going up, that means photosynthesis is happening. The opposite is true of respiration. Respiration produces carbon dioxide. Therefore, there's going to be more carbonic acid. Therefore, it's going to be more acidic. Therefore, pH will decrease. So again, in any of these experiments, a decrease in pH indicates respiration is happening, right? This is kind of a silly way to remember this, but if it helps, think about what we need for photosynthesis, right? For photosynthesis, we need the sun, right? We don't necessarily directly need the sun for respiration, but we do need for photosynthesis. And where is the sun? Well, the sun is up. So if you remember the sun is up, then hopefully that'll help you remember that an increase in pH is an indication that photosynthesis is happening. So here's how we're going to do it. You're going to have these little cuvettes. They don't actually look like this. I have better pictures. I should have put them on there. If you had you come to lab this week, you would have seen them. But it's the same concept. It's a little plastic thing that has algae beads in it and liquid, right? And this liquid that's in it includes water, but it also includes pH indicator, right? So they're just going to be sitting there doing their thing. And like I said, if photosynthesis is happening, pH is going to increase, right? If respiration is happening, pH is going to decrease, right? So you'll be able to compare these little plastic tubes filled with algae beads and liquid and see their pH, right? And every 5 minutes, you're going to measure the pH or look at it and determine the pH, and you're going to write it down here. So let's say, for example, we did this. Let's just do another one. Let's assume this had let's assume this is two different things, right? Let's assume this one is close to the light source, and this one's far away from the light source. And they both start off with a pH of 4, right? And then let's say we do the full 45 minutes, we check the pH again, and look at that. They are both a pH of 7. So if we just took the data at the beginning and the end, that wouldn't help us, because then we couldn't answer, like, which photosynthesizes better? Something close to the light source or far away from the light source? Like, we can't tell, right? These, according to this data is exactly the same. So this is why we take pH every 5 minutes. So for example, the one closest to the light source might go from 4 to 5 to 6 to 7, and then just plateau off at 7, right? And then this other one that was farther from the light might be stuck at 4 for a while. And then it goes to 5, or it's stuck at 5 for a while. Then it slowly goes up to 6, right? And then finally it makes it to 7. Clearly, this one right here on the left is the one that photosynthesized faster, right? So yes, they both started at 4 and ended at 7, but this one got up there much quicker. It's a good time for me to tell you too, there is a limit. I don't know what it is. It depends on a lot of factors. So I don't know what the limit's going to be in this particular experiment that you do, but it can only get so high and so low. Eventually it will plateau at a high level or max level, and it's not going to be 14. It's not going to be 1. I don't know what it's going to be, but the point is sometimes it will plateau. So this is why it's very important to take data every 5 minutes. Or a better way of saying that, this is why it's important to take data more than just at the beginning and the end, right? We need some middle data. That'll help us paint a better picture. So that's why we do it every 5 minutes and not just at the begging, not at the begging in the end. Let me fix this while I'm on here. And I sort of apologize if you're watching this video, you're dealing with me editing a mistake I made, but with all due respect, this is, I guess, the price you pay for, good grief. I can't type and talk at the same time. I guess the price you pay for not being physically present for lab. Anyway, back to it. There we go. Yeah. So again, no matter what experiment you're doing, this is basically how it's going to work, right? You're going to put one of these things in a particular situation, and you're going to measure it every 5 minutes to see if it's photosynthesizing or it's respiring, right? You're going to check the pH to see what it's doing. That's the basics. No matter what experiment, that's the basics of what you're going to do. So let's move forward. Those cuvettes, again, they don't look like this, right? They look a little bit different. But as far as we're concerned, it's the same thing. It's a plastic container filled with algae beads and liquid. So again, when I use the word cuvette, that's what I'm talking about, right? That's a plastic container that's filled with the algae beads and the pH indicator and all that. And again, we're going to use those things to answer these 4 questions that I'm not going to discuss again because I just did it earlier. So we won't talk about them in general, but let's talk about them individually. If you, this is a good time also for me to remind you, there's 4, as usual, there's going to be multiple experiments, but you and your team, you're only going to do one of those experiments, right? You're not going to do all of them. You're only going to do one. But as usual, the entire lab report or the entire lab, as usual, needs to be filled out, right? So you have to fill in all the tables and answer all the questions, even for the experiments you don't do. It's another good time for me to remind you, just because you're doing experiment 1, for example, doesn't mean that you can't walk around, right? You can walk around and talk to the people who did the other experiments, especially after you get your data, because we had an issue last week where the data is really confusing for one of the experiments done in the diffusion and osmosis lab. And what should have been done, and maybe it was, so I'm not saying this is to blame anybody, or I wasn't there, but what should have been done was you guys, before you left that lab, should have looked at that data and said, well, this is weird. I don't understand this, or there's something wrong with it, right? And talk to the people who did the experiment. So make sure you do that this week. It's even more important because, again, not only are you doing the lab, but you also have to do the lab report. Anyway, experiment 1. The question, again, that we're answering is, do photosynthetic organisms photosynthesize faster in more intense light? So long story short, we're going to have, we're going to have, I'll try to draw this a little bit better. We'll have a lamp, shining light, right? And we're going to have a cuvette close to the light. We'll have a cuvette farther from the light. And then we'll have a cuvette in the dark, right? That's what it calls for. However, if I remember correctly, and that little circle indicates it's wrapped up. It's shielded from the light. Anyway, what we'll also probably have, just so you know, this might be a little outdated, there might be a third where one is, so we'll have one really close to the light, one that's close to the light, and one that's farther than the light, right? That's how it's going to work. And then again, one in the dark. And you see here that there's even specifically say 20 centimeters, 25 centimeters. And when you look at the lab, it will also be very specific and tell you exactly how close. But this is your experiment, so you can do it however you want. So if you want to put it, directly under, and that's only, 5 centimeters, then you can do that. You can do 5 centimeters. And then maybe put this one 10 centimeters away, right? 10 centimeters. And maybe make that one 30 centimeters away for some reason if you wanted to do that, right? So 30 centimeters. It's up to you. This is your experiment. You can make changes. Just make sure you ask us first. Anyway, this is basically how it's going to go, right? Again, you have the lamp, you have the cuvettes. You're going to take their pH every 5 minutes. And what do we expect? We should expect anyway, the pH of that one to increase, right? All these ones that are exposed to light, we should expect them to increase. This one that's not exposed to light should be respiring. We expect it to decrease, right? So all that information is right here. This is what we expect. And we'll see. We'll see if that's the case or not, right? If we just knew it for a fact, we wouldn't need to do the experiment. So we're going to do the experiment to see if that's how it works. Another thing to mention while we're discussing this is, like I said, this one that's in the dark, it's going to be wrapped in aluminum foil. Again, its pH should be decreasing because it's respiring. However, think about this. Every time you check the pH, you have to unwrap it from the aluminum foil and expose it to light. And we're going to get that room as dark as we can, but you guys know, you've seen it. It doesn't get that dark. and you can do the best job you can, and you should, to try to shield it from light. But still, it's going to get a little bit of light. So if you notice right off the bat, like 5 minutes, maybe it starts at 4, and then it stays at 4, and it stays at 4, and you're not seeing any change every 5 minutes, then it's up to you. If you want to, you can, again, call an audible. This is your experiment. You can make some changes. You might decide, you know what? It stays at 4. The pH isn't changing. And you might think, well, probably because every time I expose it to light, it's photosynthesizing, right? In which case you might say, you know what? Let's not check the pH for the dark one every 5 minutes. Why don't we check it every 10 minutes or every 15 minutes, right? And I'll leave that up to you. And you might not even need to. Who knows? It might be that you start doing this experiment and it starts at 4, and then it goes down to 3, and then down to 2, right? It might work. Who knows? But anyway, the point being, you can see now what you should expect from the first experiment. And you can see now some of the audibles you can call, so to speak, some of the adjustments you can make if needed. If somebody was here, I would ask if there's any questions, but no one is here. So let's move forward. All right, this next one is even more simple. Same kind of concept. We have a horribly drawn lamp, right? Nice office lamp like this. Lights coming. There's a little light bulb. Got some light coming down. And we're going to have a cuvette under that light. And then once again, we'll have another one somewhere else wrapped in aluminum foil, maybe put it in a drawer, whatever. One in the light, one in the dark. You're going to take measurements, as always. Take measurements every 5 minutes, put it in here. Again, the one in the light, we expect it to go up. The one in the dark, we expect it to go down. Yeah, and again, like I said, the one in the dark, every time you expose it to light to check the pH, that is going to affect things, right? So again, if you need to, you might make the call and say, all right, well, we're only going to do this. We're going to measure it once at the beginning. I don't know, once towards the middle, and once at the end. Who knows? We'll figure it out. We'll make that decision, or you, with our help, will make that decision as the experiments go on. Another thing I should point out, and I didn't mention this in the first 2 lectures, so in a sense, you might have or the first 2 times I gave this lecture. This is sort of like the gold standard, not the gold standard. How do I want to put this? Is almost like the experiment that all the other experiments need to look to, because this is the most basic. And we've already talked about why the pH should increase in the light because of photosynthesis. And we already talked about why it should decrease in the dark. So we can look at this experiment, no matter what experiment you did, you can look at this one and see, did things happen the way we expected them to? Did this pH increase? Did this one decrease? If not, that is an indication that something may have gone wrong. Maybe the algae beads are dead. Maybe there's some other contaminant in the liquid inside those little plastic containers. Maybe you're measuring it wrong. There's so many things, right? So this is the one we're going to kind of look to. Now, don't get me wrong. If this one goes wrong and we don't have anything that's, we don't have what's expected, that doesn't necessarily mean yours is also incorrect. But it is just one of many red flags. Let me put it that way, right? You should always be looking for red flags when you're doing experiments. You're looking for stuff that's, invalidates your data. And that would be one of those things that might indicate something is wrong. Anyway, that's the second experiment. Let's talk about the third one. The third one is a little bit more complicated. In a sense, what we have here is 2 different experiments. We have a photosynthesis one in the left, right? The one in the light. We have a respiration one in the dark. So the one in the light, the photosynthesis one, is pretty simple. We'll have another horrible drawing of a lamp. All right, there's the lamp again, light coming down. But this time we'll have a little container of ice. Right? Another horrible drawing. A little container of ice. And in that ice, we'll have our little block of our little tube, our cuvette filled with pH indicator and algae beads, right? And it'll be hitting direct light and it'll be on ice. And then right next to it, in the same amount of light, we're going to put one not in the ice, right? So we'll have one at room temperature, one in the ice. And here we're just going to measure the pH. We expect the pH to increase for both of these because This is a photosynthesis experiment, right? So the question is, which one's going to increase quicker, right? Which one's going to photosynthesize more? The one on the ice or the one not on the ice, right? And whichever, your hypothesis is your hypothesis. So we'll see. And that's how we'll test that. And then the other one, again, on the other side, then we're going to do the same experiment, except we're going to do it in the for respiration, right? So we'll have this container filled with ice, and we'll have that cuvette filled with algae beads and pH indicator, also wrapped in aluminum foil, right? So it's in the dark. And then we'll have another one, right? Another cuvette wrapped in aluminum foil in the dark. And then this one's on ice. This one's in the room temperature, right? And again, whoops, I shouldn't draw that. This one's on ice. This one's room temperature. And again, we should expect, because this is respiration, we should expect the pH for both to go down. So the question then is, which one's going to go down quicker, right? Is it the one on ice or the one at room temperature? Or is your hypothesis that they're going to do it at the same rate? That's up to you, right? That's your hypothesis, and that's how we'll test your hypothesis. Some things to mention about that before you form your hypotheses, because you'll have to do that for the pre-lab. Think about when we're talking about warmth or temperature, right? What do we know? What have I taught you about temperature? Well, you know that generally speaking, the warmer something is, the quicker the chemical reactions are happening. Therefore, we should expect more to an extent, because you also know that if something gets too hot, it will denature the enzymes, right? It'll basically melt the enzymes. And without the enzymes, you don't have any of the reactions. So yes, to an extent, in general, we do expect the warmer it to be, the warmer it is, the faster it'll photosynthesize. But then you have to consider, we don't know where these, I don't know where these algae beads are from. I don't know what their species are. If this is some algae that they scrape off the bottom of an iceberg, you can pretty much expect it's probably evolved to work most efficiently in cold weather, right? Because that's what it's evolved for. So another thing from the lecture is enzymes. They all have a very specific range of temperature in which they work, and every single enzyme is different, right? They all have their own little their needs, so to speak. So anyway, keep that in mind. Then the same can be said with respiration, right? Because we're still dealing with chemical reactions and enzymes. So again, generally speaking, we expect things to happen more when it gets warmer. There's a point where it gets too warm. And also, generally speaking, there is a specific temperature range that enzymes work best at. And we do not know what kind of algae, what species algae this is, and we don't know what that optimal temperature range is. So we really are just taking a shot in the dark, so to speak. So that's the third experiment. Here's the fourth one. To me, I don't know why, but this one's the most interesting to me. Here, we're answering the question, does the color of light affect the rate of photosynthesis? And again, very simple, right? So 1, 2, 3, 4, right? We've got our 4 lamps, light bulbs, right? Shining light down. This one's shining light down. This one's shining light down. This one's white. This one's green. Not that this order matters. This one's red. This one's blue. If I can get more colors between now and then, also, by the way, we'll get more colors. But for now, this is the plan. Again, a cuvette under the white light, cuvette under the green light, cuvette under the red light, cuvette under the blue light. And again, we'll just, for all of these, every single one of them, since this is a photosynthesis experiment, we expect the pH to go up. So the real question is, which one's going to go up quicker, right? Which one of these colors is best for photosynthesis? For that matter, which one of these is the worst for photosynthesis? And we will run these experiments and we will see. One of the things I mentioned on Monday that I didn't mention today at the previous lecture that I'm going to mention now is this idea of how color works, because I'm not sure we'll have time to talk about it in lecture before you do this experiment. But when you see, for example, a plant, right? A plant is green, right? Mostly, right? Usually when you think of a plant, you should think of green, right? Because of all that chlorophyll. And here's a little thing about physics you might not know. The reason that plant appears to be green is because the pigments in that plant are absorbing all the colors, right? So white light, by the way, that's what I should have led off with that. White light is all the colors mixed in one. That's why when you put it in a prism, you can make a rainbow, right? Because it splits those lights up. But anyway, you shine a white light on a plant and it appears green to us. And the reason is because that green plant is absorbing every single color except green. Green is the color that's bouncing off and hitting our eyeballs, and then our brain says that thing is green, right? Um, right now, I'm wearing a red shirt, for example, and the reason it, not that you can see, but the reason it appears red is because white light is hitting it, right? All the colors are hitting my shirt, and my shirt is absorbing all the colors except red. Red is the one color that's not absorbing. It's being bounced back and hitting my eyeballs, right? So, and I'm not going to go too deep because I want you to be able to think about this and answer it on your own when you do your hypotheses. With that being said, which color do you think is the worst for plants, and which one do you think is the best for plants? And again, think about the nature of colors, right? My truck is red because it's absorbing all the colors except red, right? My wife's eyes are green because they're absorbing all the colors except green, so on and so forth. And that's it, right? So now we're finally done talking. Oh, how about this? The first word for attendance, because again, you're gonna need to prove you watch this either for credit for lab, or some of you go-getters might be doing this for extra credit, which would be even better. But anyway, the first word for attendance, let's say, is going to be beads. Right there. Beads is the next word for attendance. All right, moving forward, let's talk about these other ones here. All right, so again, now we're done talking about the specific experiments and how we're going to answer those questions and things like that. Now let's look at some random notes, some things to keep in mind as you do your experiments. I want to make this very clear, and this will help you when you do your pre-lab, but the purpose of these experiments is not to see how the pH changes, right? This is not a pH experiment. We are just using the pH as a tool to measure the rate of photosynthesis and respiration, right? This is not about pH. pH is just a tool. Just like last week, you did a diffusion and osmosis lab, and you had to use a scale to weigh those artificial cells. But that wasn't a lab about weight, and I've done labs about weight. There are labs about that in the physics class, but the lab wasn't about the weight of a cell, right? That was just a way of measuring whether or not water came into the cell or something left the cell, right? It's just a tool. Same thing here. So yes, we're using pH, but please don't focus on that, right? It's not like it's not like the question we're saying is, all right, if we put one cuvette in white light and one cuvette in blue light, which one will change the pH the quickest, right? That's not technically what we're doing, right? It's more like we'll put one in white light, one in blue light. Which one is going to photosynthesize the quickest? It just so happens the pH is how we're going to measure it. And again, like I mentioned earlier, if we had all semester to do this, we could grow a plant. Right? We could grow plants under these lights. And in which case pH wouldn't matter, right? In that case, our tools to measure growth we could measure how tall the plant is, or how much it weighs, or count the number of leaves. But so again, pH is just a tool. And I've already mentioned this, but here it is in writing again. No cuvettes in the dark should have an increase in pH, right? If it's in the dark, it should be respiring. It should be producing carbon dioxide, which produces acid, which would decrease the pH, right? If it's in the dark, it should not increase. Likewise, none in the light should decrease, right? They should be increasing, because in the light, they should be photosynthesizing, using carbon dioxide, reducing the carbonic acid. So if any of those things happen, and that's kind of what I was alluding to with that second experiment, we can also look back to that second experiment as kind of to see if there's any red flags or not. As a matter of fact, I'm going to scratch this word because I don't necessarily stand by what I wrote this time. If these things happen, I wouldn't say your results are invalid, but I would definitely say we have a red flag, right? So if there's a bunch of red flags, then you might say and then again, it depends. Every situation is different, but that's definitely a red flag. So it might be that your results are invalid. It might be that it's just a red flag, but think about it and talk to us about it, right? It's not up to you as a freshman or someone taking a 100 level biology course to be able to decipher the results by yourself. We're willing to help. So let us know, but that's just another random note to keep in mind. Some more random notes is while performing these experiments, and I've alluded to this already too, but think about how they could be performed in a better manner, right? So as you're doing this, think, man, and I'll give you one, so you can't use this, and you're going to have questions about this. Well, no, I guess you could use this, but there's going to be more. So I'll give you this one, and then you can think of some other ones. Like I already said, every time you check the pH of the one in the dark, you have to remove it from the aluminum foil, exposing it to light. So that's one of the ways that it could be performed in a better manner, right? So in this case, you don't necessarily need a solution, but you at least need to identify the problem, right? So you could say something like, this experiment would have been improved if I could find a way to measure the pH of the cuvettes in the dark without exposing them to light, right? And that's just one of the many. So think about that, and we'll be talking about it as the experiments go on, and you will be asked that on the lab worksheet as usual, and you will have to include a paragraph of that in the lab report. So make sure you do that. Think about that. Ask questions if you need to. And then again, I've already mentioned this too. Think about different experiments that could be performed to answer the same question. So you're basically going to need to write a description. You need to come up with an experiment design to test the same hypothesis, but using a different experiment. So again, for example, and you can't use this example. Well, for example, in this lab, you might do the experiment with the colors of light. And in this lab, you used algae beads and placed them under different colors of light. And you measured the rate of respiration by checking the pH every 5 minutes for 45 minutes, right? That's a basic description of that experiment. So that you're going to need to do another basic description of another experiment you could do. Because in real science, when you do an experiment and it supports your hypothesis, you're not done usually. Then you find another way to test your hypothesis. And here's a good example. I'll give you this example because this is one that you should not use. So I'll tell you one you shouldn't use, and I'll explain why you shouldn't. And I've seen people do this before. People will then say, because this is a question you'll be asked, they'll say, we could do a different experiment where we put algae beads in the lights, blah, 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 and we measured the pH for, I don't know, 60 minutes, right? They only changed the minutes. Like, that's really not a different experiment, right? So come up with a different experiment. Don't just do one small thing like, oh, we could just do it longer. That's not going to work. This is especially true because you'll see with your data, like I mentioned earlier, sometimes the pH plateaus, right? It might hit, I don't know, I'm just going to throw out a number. I don't know. It might hit 8 and never get any higher than that. It might hit, I don't know, 4 and not get any lower than that. I don't know what it is, but there is a limit. So there is no, and we'll probably hit that limit in these experiments. So if the pH, for example, for one experiment goes up and up and up, and then it just hits this, let's just say that's at the 30-minute mark, right? And then, here you are at 45 minutes. There's no need, right? What use would it be to keep doing that experiment? We've already maxed out, right? So anyway, again, think about different experiments you can do. You'll need to write a basic description. As always, you don't have to you need to come up with the idea on your own. But then if you need help on how to write the experiment design, the basic description of the experiment design, then come to us for help. But yeah, think about different experiments you could do. Oh, another example of what not to do I've seen before is somebody said something like, instead of testing the pH of the algae beads, you could test the pH of plants. And that clearly indicated to me that they don't understand the experiment that they did, right? Because you cannot test the pH of plants the way we described it, right? The only reason we could test the pH is because those algae beads are in liquid. And as those photosynthesize, they're taking carbon out of that liquid. Therefore, it's getting less acidic, right? We can test the pH. We're not really testing the pH of the beads. We're testing the pH of that liquid. Likewise, when it respires, it's putting out more carbon dioxide into the liquid. And that's creating an acid in the liquid. And therefore, the liquid's pH is decreasing, right? So you can't just say, we'll take the pH of of a plant instead, right? That doesn't work. So think about your answers and come to us if you need help. So that's it on the experiments themselves, right? How to do the experiments. And you can see now why I wanted to break this up into 2 weeks. So this way, next week, after a brief discussion, when we all start, then you can just hit the ground running and get to work. So here's the other part. Let's talk about how to write a lab report. So the first question, before I tell you how, let's answer the question why, right? Because for the most part, none of you are biology majors. You might be thinking, why do I need to write a lab report? This has nothing to do with my major. I'll probably never have to write a lab report again in my life. And that's probably true. And the first, most basic answer is this. It's part of the course, right? I didn't make that decision. Somebody above my pay grade decided, hey, everybody in a 101 class needs to write at least one lab report. It's part of the scientific literacy. It's just the way it is, right? Another part, and this is the part that is in my control, and I happen to agree with this. You can't really write a lab report unless you truly understand the experiment. As you've noticed, hopefully not many of you, but surely some of you, unfortunately, have noticed that you can just kind of attend lab and write down the data and kind of use that data to answer some questions. So you can get through a lab and get a decent grade, maybe even a great grade, given that I'll allow you to correct your mistakes, but it doesn't require a great understanding of the experiments you did to do the labs, right? But to do a lab report, you truly need to understand it. And in a perfect world, I would have you do a lab report for every single lab, but that wouldn't be fair to you. And it's only a 100 level class, and I wouldn't have time to grade it all. So it is what it is. And then the other part that's, I think, more important on a practical level, again, because none of you probably are going to be scientists and doing the lab reports in your real life. But hopefully what you will do as an educated citizen of the world is hopefully you will read research articles. And basically a lab report is like a Cliff Notes version of a research article, right? So by giving you experience on writing a lab report, you're kind of getting an experience on writing a research article, right? In a sense. So hopefully this way you'll be a little bit more familiar with how research articles work. And this is important. Again, this is probably bigger than learning biology itself in a way, to be a critically thinking citizen of the world, right? You should look things up. When someone says, you got your Uncle Bob saying you should not take that vaccine, but then you've got, your cousin Jimmy who says you should take that vaccine. Well, do some research, right? Do some research and look up some articles. And again, to understand those articles, you need a little bit of background knowledge in biology, which you're getting in this course. And it helps to understand how research papers themselves work. And that's what you're doing in this situation. That out of the way, let's talk about some other stuff. Do not use the internet. Let me be very clear on this, which is why I've got this little star here. What I mean by that is when it comes time for you to write your lab report, do not Google how to write a lab report, because when you do that, you're going to find all kinds of answers. Because just like lab reports, or just like, research articles, right? Every single journal that publishes research articles, they have different requirements of how they want it written. Same here, right? There's different ways of writing a lab report. So I'm going to tell you how I want it written, and that's how you need to follow my guidelines. Another one, and I've seen people make this mistake here, to make things a lot easier for you. You're only going to write a lab report about the experiment you did, right? So there's 4 experiments that are going to be done on the regular old lab worksheet. You got to answer all those questions, write down all the data. But when it comes time to actually write the lab report, you only write about the one that you did. But that being said, if you want to write a lab report about the other experiments, actually, I would recommend it. You could do those first as practice, and I will count that as independent work, right? So you can write a lab report on the other ones for independent work. And again, great practice before you write your actual lab report. Some other things, and this is easy points, just make sure you do this and you won't lose points for it, but it needs to be past tense and in first person. So you'll say something like, in this experiment, I wanted to see how temperature affects the rate of photosynthesis and respiration. And this experiment, the first thing I did in this experiment was put the cuvette directly under the light, right? So it's first person, past tense. Another thing, again, just something you need to do, easy points if you do it. Otherwise, you lose points for nothing. Make sure it's double spaced. And I don't have this on here, so I'm probably not gonna deduct points for it. But let's just go ahead and put it this way. You know what? No, never mind. Since you're doing Google Forms, whatever font and size it's already in when you pull it up, I'm fine with that. No need to make changes. Here's another big one too. There is no minimum. I always get that question. How many pages does this need to be? How many words? There is none. And in my opinion, the shorter the better. Generally speaking, the shorter the better. You just give me the information that's required, and you don't need to go on and on. And this is not like what's the word I'm looking for? You're not writing to be entertaining. So you don't want to say something like, the first thing I did was grab the cuvette and very gently put it under the light. And then I sat and thought, what should I do next? And I decided I should grab the next cuvette. So I did. And I put it very carefully farther from the light. You don't need to be descriptive, right? Just straight to the point, which should make things easier for you and also easier for me as I'm grading them. You're going to turn them in the same way you would a lab or independent work, and what I mean by that is you're going to do Google Docs. So let me back out of this and show you what I mean. Oops, excuse me. I have an example pulled up ready to go. Where is it? There we go. So this is the sheet you're going to get. Just like a lab. You know how you get a lab worksheet? Well, I'm going to share this with you. It'll probably be slightly different. But anyway, I'm going to share this worksheet with you. You're going to hit the share button. You're going to make sure it's shared with me. Of course, let me back up a step. You're going to get this document the same way you would a lab. The first thing you want to do is go to File, make yourself a copy, right? So then you have your own copy because you can't edit the master copy. Make yourself a copy. And then, of course, you're going to share it with me, right? And then you can see here, and I'm going to talk about this here in a second, but everything you need to do line for line, it's there, all right? Is there an abstract? Okay. Is it a brief summary, right? And you just go through this line for line and make sure you follow everything. But anyway, my point is, here's what you need to do, right? There's your grading rubric, right? Grading rubric. And right here, as it says right here, actually, I'm going to make this a little bit larger. And then start doing your lab report right there, right? So that's how you're going to turn it in. Just like you would independent work or a lab. Make yourself a copy, share it with us. All right. That being said, let's get back to this PowerPoint. Let me back up a second. Okay. Anyway, another thing, and you're going to be reminded of this, but your due date is 2 weeks after you get your graded lab back. So say, for example, you're in the Wednesday lab next week, and you're done with it on Thursday. And I grade it on Saturday. And on that day, when I give you your grade, I will say your due date for your lab report is 2 weeks from today. Of course, at that point, I'll give you the actual date. Actually, I might as well look it up now, just out of curiosity and to be a little bit more specific. Like I said, if I gave it to you, grade Saturday is the 24th, right? So if you get your grade back on the 24th, in the comments, I will say, your lab report is due in 2 weeks on, so that would be what, February. So not March 2nd, but March 9th, right? So it'll say your lab report is due in 2 weeks on March 9th in this for this example. Anyway, another thing I want to point out too, this is very important. There's a lot of the stuff you can write before you even do the lab report. You can start writing the abstract, and I'll talk about that when I get there. You can write the entire introduction. You can write the entire materials and methods, but it might need to be edited. So anyway, yeah, keep that in the back of your mind. Basically what I'm saying is you don't have to wait until you get your lab graded to start, right? You can go ahead and start now, but 2 weeks after you get it back is when it's due. All right, let's move forward. As usual, as everything you write should be free of grammatical, spelling, and typing errors. I'm not very picky with that. So if I'm deducting points for it, you can guarantee it must be pretty bad. Another thing is, again, you're using Google Docs to write this, and when you misspell something or have grammar errors, if they're they tell you, right? It'll highlight it or give you a little red underline. So just look at your document. If you see any red lines, fix it. It should make sense. This is a big one. This is a good meter stick here. You should be able to give your lab report to somebody who's not in this class, who's never had this class, who has no idea the lab you did. Like, you should not be able to say nothing to them. Give them the lab report, have them read it, and understand what happened, right? There should be enough information where they're like, I get it. I know what you did. I know what these results mean, right? That should be your biggest meter stick, right? I mean, yeah, I'm going to give you a rubric, but that's the big one. If it doesn't, it should be able to make sense to somebody who's never done the experiment. It should be accurate or correct. For example, like, again, people do make mistakes, and they'll say something like in the introduction, which I'm about to talk about. They may say something like, in your introduction, you have to describe the whole pH thing that I talked about earlier. And someone might say, well, when photosynthesis happens, it produces carbon dioxide, and that makes the pH go down. I'm like, no, hold on, back up. When photosynthesis does not produce carbon dioxide, it uses it, right? So be careful for stuff like that. Be extra careful, as a matter of fact, because if you write something like that, spell check isn't going to check, it's not going to notice that. So be very careful. It should be precise. I don't really have time. I don't want to waste your time getting into that discussion. Maybe I'll discuss it when we're in lab, but it's also more important for other types of experiments as opposed to this one. There's not much precision involved in this one. It should be succinct. This kind of goes along with what I was saying in the previous slide. There is no minimum words or maximum, whatever. The idea is to be to the point, right? Don't be trying to make it entertaining, like when you're at the end. So my results surprisingly showed me that my hypothesis was correct, and I was very happy. You don't need that, right? It's not a narration. It's not supposed to be entertaining. It's just to the point. It should be clear that you understood the experiment. That's another big one. This is like the other big one that I circled. One, it should make sense to somebody who has no idea what it, has never had the class and has no idea what you did. It should make sense to them. And to me, when I read it, I should understand. I should be clear that you understood the experiment, right? So when you write it, that's the biggest thing I'm looking for, is to see if you understood the experiment. And it's hard to give you a lot of, I've seen so many examples throughout the years of like, what did they write? Like, I don't understand I don't think they understood the experiment. So yeah, be careful of that. These kind of go together, actually. A lot of the times when people write things that are not accurate or correct, that usually indicates they didn't understand the experiment too. So again, the grading rubric will be shared, not in Google Classroom. This is an old thing. I'll be sharing it via my online as usual. Actually, I'm going to send it hopefully today. So again, I just showed you a couple seconds ago. You're going to have that document, right? You're going to make yourself a copy. You're going to share it with us. And I've already shown you, but this is what the top of it looks like, right? You already saw it. That's what some of it looks like. So again, if you just go line for line, it's hard to mess it up. Step one, is there a title? Well, I better give it a title, right? So on and so forth. So here's what it's going to be broken down to, right? Your lab report is going to be broken down in the following sections. Each one of these we're about to discuss separately, and also each one of them is going to be labeled. The first thing, well, before we talk about it, right, we're going to have a title, then an abstract, then an introduction, materials and methods, results, discussions, figures and tables, literature cited, all in this order. Don't mess the order up. So first thing, let's talk about the title. Again, you're going to be performing, or not you, but as a class, we will be performing 4 experiments, but you yourself are only going to do one of them, and that's the one that you're going to write about. And on that note, your title needs to indicate which one you're writing about. So a title that will lose points for you is something that says photosynthesis or photosynthesis respiration lab. Right? That doesn't tell me what your experiment was about. That's the name of the lab. That doesn't tell me anything. And then you can see here, that's basically in writing what I just said, right? Photosynthesis and respiration. Not a good title unless you were writing a lab report about all of them, which you're not. So again, you'll be specific, like the rate of photosynthesis at different colored lights, if that's the one that you happen to be writing about. So that's the title. Very simple. Abstract. Let me say this. Basically, the abstract, and this is especially true when it comes to real peer-reviewed journals, it's like a brief summary of the whole thing. And a lot of times, even scientists very often only use the abstract because if you, I don't know how to put this, maybe not so much as a scientist, but if you're writing a paper, and you guys do write papers for independent work, but if you were writing a more in-depth paper and using, and I was like being really strict on the sources, you would look up peer-reviewed articles, and very often, like you would just, like if you made a statement and you needed to back it up, you would back it up by citing a source, and that information that you needed was probably in the abstract. So if you said something like carbon dioxide levels in the atmosphere decreased by 3% from the year 2019 to the year 2020 as a result of COVID-19, whatever. You would, there's somewhere out there, there's a paper that you read, and you probably just read the abstract, and it gave you that information, right? So anyway, that being said, let's talk about your abstract. As always, everything, it should be labeled. So literally, you're going to write the word abstract, and then, go under that and write your paragraph. That's going to be your abstract. It should let the reader know what the rest, if they want to read the rest. So again, not that this is very important for you to be writing a lab report, but just so you know, again, for example, if a scientist is looking to answer a question that they don't have an answer to, another thing, what they're going to do is something called a lit review, a literature review, which is where they look to see what other scientists have done and what they already know. And no one has, well, I shouldn't say no one, this is kind of, but it's not really practical to go read just a bunch of articles, right? People don't have time for that. So what you do as a scientist is you read the abstract, like, yes, this is more along the lines of what I was looking for. And then you'll know that you want to read their article and see what their materials and methods were and exactly what the results were, right? Because that's more along the lines of the research that you're doing. But anyway, moving forward. Okay, so again, your abstract should be a brief summary of what the paper is about. In other words, it should include the question being answered in the lab. And I'm going to use the last one. Let me give you an example. So it could say, in this experiment, I wanted to answer the question, does temperature affect the rate of photosynthesis and respiration, right? That's the question being answered. Then you give a basic description of how you did it, right? So again, using this as same example. In this experiment, I wanted to answer the question, does temperature affect the rate of photosynthesis and respiration? Here we go. To answer the question, we put cuvettes filled with algae bead and pH indicator and put them in different conditions while measuring their pH to determine the rate of photosynthesis and respiration, right? That's very basic. I didn't say exactly, we put this one on ice and put this one in aluminum foil, right? Just basic description of what it was. And then you give the basic results. So again, using the same example, you would say, again, this is the question I wanted to ask or answer. This is basically how we did it. And what we found is, yes, temperature does make a difference because, and again, I'm just using this as an example. We found that colder temperatures are better for respiration and warmer temperatures are better for photosynthesis, or whatever the case may be, right? You don't give you don't say, this one, the pH went from this to this, and this one, the pH went from that to that, and here's our results. But, you don't have to, just the basics. So again, and I know I'm going on and on about this, but really, ironically, as long as I'm talking about this, the idea here is for things to be nice and short and brief and easy for you. But yeah, that's the abstract. Next comes the introduction. This section gives enough information that the reader can understand the rest of the lab report. And you'll see what I mean here in a second. As always, it should be labeled. So you have the word introduction, and then you'll go down and start your paragraphs. It should give enough information about photosynthesis and respiration that the reader will understand the rest of the lab report, right? So just the basics, right? At the very least, you should discuss what I've talked about, which is, photosynthesis uses water and carbon dioxide to produce oxygen and glucose. And then that glucose is used in respiration in the presence of oxygen to produce um, carbon dioxide and water and ATP, right? Just the basics, right? Just the basics. Now, this is one of those times where I guess you could get a little bit deeper, and I'm not going to penalize you if you get very deep, but honestly, I'm not looking for that. I'm not trying to determine if you are very familiar with photosynthesis and respiration, right? We'll have questions about that on the exam. But at the very least, you definitely need to talk about carbon dioxide. Think about it, because you're going to need to explain the whole pH thing, and you can't explain the pH thing if the reader doesn't already know that photosynthesis uses carbon dioxide and respiration produces carbon dioxide, right? At the very least, you need to include that. You need to include the question you're answering. Yes, I know this seems redundant. Get used to that. There's going to be a lot of redundancy. So yes, in the abstract, you already included the question, but now you're going to do it one more time. So once again, you'll say, in this experiment, I wanted to see if the color, if the temperature affects the rate of respiration and photosynthesis. And then what comes after the question in the scientific method? The hypothesis. So then directly right after your question, you need to write your hypothesis. So again, question. In this experiment, I wanted to see if temperature affected the rate of photosynthesis and respiration. I hypothesize that temperature does affect the rate of photosynthesis and respiration. Or I hypothesize that it does not. Or I hypothesize that it'll affect photosynthesis but not respiration. Whatever it is, right? Tell us what, after you restate your question, then state your hypothesis, and then include at least one sentence about why you chose your hypothesis. Right? So this is basically what I'm saying is you can't just make a wild guess. You need to tell me why you think temperature is going to affect photosynthesis and respiration. Why do you think the color of the light will or will not affect the rate of photosynthesis, right? You need to have a logical answer. You will lose points if you do not include an explanation. What you will not lose points for, generally speaking, don't hold me to this, you will not lose points if your reasoning. Let me take that back. That might be the thing. If your reasoning is too off the wall, you will lose points, right? So if you say something, and we've already talked about this earlier, if you say something that's inaccurate, you'll lose points, right? So if you say, I hypothesize that plants will photosynthesize better in the dark because photosynthesis requires a lack of sunlight and cold temperatures, then you would lose points for that, right? This is just not a true statement. So think about your hypothesis. Explain why you have your hypothesis. Speaking of which, it's a great time for me to remind you, we have separate experiments, right? So say, for example, you were doing, let's say you're doing something. Let's say you're doing the third experiment that is all about temperature, right? A better one. Let's say you're doing the fourth experiment, which is all about the different colors of light. Now, in your brain, if you think maybe perhaps white light is more intense than red light, drop that thought, right? We already have a separate experiment that deals with light intensity. Likewise, if you think, I don't know, red light is warmer than green light, and again, don't include that for your explanation because we already have an experiment that deals with different temperature, right? So your reasoning for your hypothesis needs to be completely clear and separate from any of the other experiments, right? So if you're not doing the experiment about light intensity, you shouldn't even be thinking about that, at least for your hypothesis, right? Drop that thought. If you're not doing the experiment on temperatures, drop that thought. If you're not doing the experiment on different light colors, drop that thought, right? Anyway, let's move forward. Again, we're talking about what needs to be written in the introduction. And also, by the way, I would also recommend writing it in this order. But anyway, after you've given your question and you've provided your hypothesis and your reasoning for your hypothesis, then again, you need to go back and explain. All right, now that we know your question and we know your hypothesis, then the question is well, how are we going to test it? So you can just really briefly say, to test the rate of photosynthesis and respiration, we looked at the change in pH. And then you just explain. I'm not going to waste your time or my time explaining again, but all the stuff I mentioned earlier about carbon dioxide going into the water, producing carbonic acid and making the pH drop, that's what you need to explain again. Because like I said earlier, or like it says right here, enough information that the reader can understand the rest of the lab report. So that way later, when you start talking about in the next section, materials and methods, when you start talking about, you check the pH, if the reader read this first, they'll know why you check the pH, right? So they're not going to be lost in the sauce. They'll say, oh, okay, I understand why I know why they check the pH. Likewise, and this helps you out, since you already mentioned it here, this is one of those times where you don't need to be redundant, right? So when you get to the the materials and methods section, again, you don't need to talk about why you're checking the pH. You've already done that in the introduction, right? You just say, I'm doing this. I check the pH every 5 minutes, right? Anyway, so on and so forth. Let's keep moving. The second word for attendance is going to be report. Anyway, and I should have mentioned this bullet point should have been with all the other ones. I don't know how I messed that up. This bullet point, at least this part of the bullet point could be applied to everything. Make sure you only have the stuff that's supposed to be in there, right? So for example, and I should have mentioned this earlier, in the abstract, you shouldn't talk about specifically, you shouldn't write a whole paragraph about how pH, changes, how respiration and photosynthesis change the pH, right? You don't want to go into detail. You don't want to, in the abstract, talk about how photosynthesis and respiration work in general, right? So again, make sure you only put things that are supposed to be there. Anyway, specifically to the introduction, again, don't include a lot of things. Don't include anything that shouldn't be there. In this case, some things that should not be there on specifics on how the experiment was performed. Like, yes, and this bullet point, you do say, we used pH to measure the rate of photosynthesis and respiration. Then you talk about the chemistry behind that. But you don't get specific and say, I put this pH under this light at 5 centimeters, and I put this one at 10 centimeters, and I put this one at 15 centimeters, right? There's no specifics, right? And also another thing, right here, you don't talk about the results, right? So you talk about your hypothesis, but you don't give results at all, right? So this is like, here's the basics, right? Just the basics. Here's what you need to understand the rest of the lab report. So that's it for the introduction. The next one's pretty simple, materials and methods. This section should give enough information that the reader can perform the experiment from reading it. This is so easy. That should be your measuring stick, right? Again, just like the whole thing, the whole lab report should make sense to somebody. This particular section, you should have enough information where you could give it to a student and say, here, I need you to do this experiment. And that student should be able to take it. They should know exactly what they need and know exactly what they need to do, right? So you should include everything you do that the person needs to do, right? So again, just as a crazy example, you do need to be very specific, but obviously the point here is for someone else to be able to recreate it. So for example, you don't want to say, first I picked up the cuvette with my left hand, and then I looked at it with my right eye, right? None of that matters. You think about when you're doing it, think about the details. Yes, you want details enough for somebody to recreate what you did, but there's only so many details you need to give, right? So for example, there's one last example, then I move forward. You're going to be using lamps, right? For every single one of these experiments, you're going to use lamps. One thing some things you do not need to include on the materials and methods when we're talking about what you did, is you don't need to say, I plugged in the lamp and then I turned it on, right? So it's just a given that the lamp is on. And for that matter, for example, here, this bullet point is talking about everything used. I guess, again, you will say that you used a lamp, but you don't necessarily need to say it had a light bulb, right? That's kind of a given. It had a light bulb and a cord and a switch. Whatever. Used a lamp. But don't get me wrong. If you did the ones with different colors, you will mention, I used a white lamp, a blue lamp, a green lamp, and a red lamp. But But anyway, the reason I get at this, a common mistake I see is that a lot of people forget to mention the algae beads and the pH color guide, right? So a lot of times we'll say we had We used cuvette. We put cuvettes in intense light, and then we put cuvettes in less intense light. That whole time, though, and then never mentioned anything about what's inside the pH. Now, remember, you have to assume whoever's reading this, assume it's not me in a way. They need to know what's in there, right? We had cuvettes filled with algae beads and uh, pH indicator, and we compared it to a pH color guide, right? You have to have all that. And excuse me, I have to back out of here again because this is trying to stop recording again. And I need to pull that up. Excuse me. There we go. Start right there. Here we go. Stay in the call. I apologize for this being so long, but again, not to be rude, but when you miss the lab, you pay the price. And that'll be the next word for attendance, by the way. Price. And we are back at it. The next word for attendance is price. Okay. Anyway, back at it. Materials and methods. Here we go. So again, I basically said this. You should include everything that you used and everything that you did. And again, this first bullet point, that's what you're looking at, right? Someone should be able to read it and understand and be able to recreate your experiment. What you should not do, especially now, because back in the day we had an actual physical lab manual that like pages, paper. Now it's really easy to copy and paste. You don't want to do that because I want to remind you, this has to be first person past tense, and your lab manual is not written in first person past tense. So don't do that. And then finally, here's another thing. I mentioned this earlier. These are your experiments. You might need to call some audibles. You might make some changes. Maybe you'll have some extra cuvettes or an extra colored light or whatever the change may be. You can make changes, but you make sure that in your lab report, you write about what you did, not what the lab told you to do, right? If the instructions told you to do one thing and you did something slightly different, that's fine, right? It's okay to do that, but you have to write about what you did, not what you were supposed to do. And again, this goes back to what I mentioned earlier. The introduction gives enough information. So when you're talking about, for example, you should mention, I recorded the pH every 5 minutes. You don't need to say why. I recorded the pH because if the pH goes up, that means photosynthesis is happening. If pH goes down, that means, you don't need to explain that. You already explained it, which sounds kind of silly, I know, because some of this is admittedly redundant. We're going to talk about that again here in a second. But some of it is not redundant. So that's it for materials and methods. Let's talk about results. Now, this is where it gets, I don't want to say interesting, but yeah, slightly interesting. I've already been training you to do this. So in all the labs you do, first you have a table. It's nothing but your data. Sometimes I have you do a graph, of that data. And then you have to answer questions about that data. And the first set of questions you always answer about that data is just simply, it seems redundant, and it is for a reason because you're doing it here. You're basically going to take the information that's in that table and put it in paragraph form, right? Yes, it's right there in the tables or the graphs, but now you need to write it in paragraph form. You're going to write sentences about it. At the very least, no matter what experiment you do, you need to say the starting and ending. You need to write the starting and ending pH of every single cuvette, right? So the pH for the cuvette and the red light started at 4 and ended at 10. The pH for the cuvette and the green light started at 4 and ended at 5, whatever it is, right? At the very least, you're going to do the beginning and ending. Actually, let me do that as an example really quick. Let's just say you were only doing, I don't know, red light and blue light, right? And again, we've seen something like this before. So let's say they both started at 4. They both ended at, I don't know, 8. Actually, no, let's use this one. One started at 4, one started at 9. Let's assume this is the experiment you did that only had red light and blue light, and that was it. In this case, if this was your experiment, you'd be done. That right there is the results section. You're going to put this in paragraph form. You could just say the cuvette and the red light started at 4, ended at 8. The cuvette and the blue light started at 4, ended at 9. Boom, that's done, right? That's all the pertinent information. You don't need anything else. Now let's do another example. Let's say red, blue, and green let's say this is the experiment you're doing. Aha, there we go again. This time, 2 of them ended at 4 or started at 4 and ended at 8, right? They both ended at 8. So in that case, this bullet point will not suffice. In this case, you also need to include which cuvette pH increased or decreased the quickest or the most. So again, going back to that example I had before, and I'm changing it a little bit, but I don't know, red and green, those lights. And I'm going to abbreviate this to save your time and my time. Let's say this one starts at 4. This one goes to 5, 6, 7, 8, right? And that's the end of your experiment. Again, that's obviously going to be longer than that. And this one goes to 4. That's a really bad example. Sorry. Oh, yeah, we could work with that. 8, 8, 8, right? And this one goes to 4 and stays at 4. Then it goes to 5, then 6, then 7, then 8, right? So now, in this case, you can't just do this first bullet point. You can't just say this one, the one in the red started at 4, ended at 8. The one in the green started at 4, ended at 8. You will say that, but obviously in this case, that's not enough. Then you need to go on and do this second bullet point, if applicable. And in this case, it is applicable. This time you'll say, again, the one in the red started at 4, ended at 8. The one in the green started at 4 and ended at 8. The one in the red, or you could squeeze this into one sentence if you want, started at 4 and ended at 8 in a matter of, let's see, 5, 10, 15, 20 minutes. The one in the green started at 4 and didn't reach 8 until 5, 10, 15, 20, 25, 30, 35, right? So in that case, you do need to do this bullet point and explain which one got there quicker, right? And again, I'm just using photosynthesis as an example in this discussion. That's why I keep saying increased, right, and got there quicker. But again, if you're looking at respiration, then we're looking at how quick did it decrease. But anyway, that's that. And then finally, there's one more instance, if applicable, that you need to write about it. If one of them didn't change at all, right? If you had a cuvette that didn't change, and again, that seems a little redundant because right here, when you're doing this first bullet point, which no matter what you have to do, right? So if you said this cuvette and started at 4, ended at 4, then once again, you'll say it one more time, but your sentence will specifically specify that, right? So you say there was one cuvette that didn't change, and it was the one in the green light or whatever it may be. So that's the results. Again, you're just putting the information that you have in your tables and your graphs, and you're putting it in sentence form, right? I know it seems redundant, but it's all for a reason. Another thing you should do, and I've been doing this verbally as I've been giving you these examples, you don't want to say, I put cuvette number 1 went from a pH of this to this. Cuvette number 2 went from a pH of this to this. You don't want to do that, right? You want to describe it by its condition. The cuvette closest to the light started at this pH, ended at this pH. The cuvette farthest from the light started at this pH, ended at this pH, right? You want to describe them not by their number, because that's not going to mean a thing to the reader, but describe it by its condition. And here's the big one too, and people mess this up a lot. You don't use the word photosynthesis or respiration, not even once in the results section. This is straight up the results. This is just saying this is what the pH did, right? This is how the pH changed. Later in the next section, then you're going to explain what that means. Like, what does it mean that the pH went from this to this? Oh, that means photosynthesis happened. What does it mean that respiration went from this to this? Oh, that means respiration happened, right? Whatever the case is, that's going to be the next section. So in this section, again, most likely you could probably just get away with this first bullet point, but depending on your situation, you might have to do some of these other bullet points. But the point is, it's just you're putting it in paragraph form. We're almost done, guys. Next is the discussion, and I've already alluded to that, right? So as you already know, the discussion is going to be a lot like the results, except we're kind of explaining the results, right? This section, the discussion section, explains the results section, right? So the results just gave, here's the change in pH. Now you're going to explain to the reader what that means, right? And again, you've already alluded to it in the introduction. You already talked about how an increase of pH means that photosynthesis happening and a decrease, right? You've already said that before, so you can be pretty quick about it. So once again, you'll explain the results, but this time you'll do it while addressing your hypothesis, right? So you could say, again, the cuvette and the red light went from this pH to this pH, and the cuvette and the green light went from this pH to this pH, meaning that this one photosynthesized faster than that one, right? So again, you're restating the results, and I know that's redundant, but this time you'll also follow it up by giving the big picture. You'll say the same thing again, but then you'll say, and that means the cuvette in this color photosynthesized the least, and the followed by this one, followed by this one, followed by this one, which photosynthesized the most, right? So you're going to explain it that way. And then again, I know this seems redundant, but you're going to, hold on, excuse me. Yep. Again, you're going to readdress your hypothesis. So yes, you said it in the introduction, but now you're going to do it again, right? So you explain those results again. The pH did this, the pH did that. That means this thing photosynthesized faster than that thing. And then you go back to it, right? And the question I was trying to answer is, does light affect the temperature? Does light affect the rate of photosynthesis? My hypothesis was blah, blah, blah. And yes, the data does support it. Or no, the data doesn't support it. Yeah. Now, this part right here, use the data to explain how. You could do, yes, somewhere in the discussion you should do that. But in my opinion, it should be either right after you restate your hypothesis, or like I said earlier, you're going to kind of say when you're doing your results, right? So again, just as a quick example, the cuvette and the red light went from this to this, and the cuvette and the green light went from this to this. Therefore, the cuvette and the green light photosynthesized more. The question I was trying to answer is, does light affect the rate of photosynthesis? My hypothesis was this. As you can see, my hypothesis was supported because the red light was better for photosynthesis, whatever it may be. Here's where it gets a little bit trickier. If the data did not support your hypothesis, right? So if your hypothesis was wrong, then you need to explain why you think it was wrong, right? So was your hypothesis wrong? Were you like, I see it now. Of course, plants need light to photosynthesize, right? Then you can explain that. Or if you think there was just something wrong with the experiment, if you still think your hypothesis was correct, like, the data does not support my hypothesis, but I think this is why, right? And another thing, just a little side note, because you will lose 1 point for this, and this goes through the whole lab, the whole course. People make this mistake, so I want to bring it up now. You're not proving anything in this experiment. You are not proving anything in these experiments. There's not enough data for you to prove anything. You're either supporting your hypothesis or you're not supporting your hypothesis, right? It's not proving anything. Anyway, that's going to be one paragraph. The next paragraph, and we've already talked about this, you've already been trained to do this, you've been answering questions like this through all of your labs, you should address the limitations of the experiment, right? So for example, using this, if you do an experiment that suggests that algae photosynthesize better at more intense light, does that mean there's no limit to the light intensity, right? So, and now you can't use this example because I've used it. So you might say, like, well, according to my data, the best distance for light intensity is 10 centimeters. And then you would say something like, but we didn't get any closer to 10 centimeters. Or, but what if we used a brighter light, right? So you would talk about how there's limitations. And another one, sorry, I've already mentioned it, so I'll mention it again. And this one you should mention. This is the one that I'm giving you. We're talking about the limitations to your experiment, right? You could say every time I checked the cuvette wrapped up in aluminum foil, and I exposed it to light, and that is a flaw in the experiment. And as always, during the experiments, we'll be walking around, we'll be talking about the flaws in the experiment. And then finally, your last paragraph, and we've already talked about this too, you should discuss other experiments you can perform. So just like any other peer-reviewed, well, it's kind of a stretch, but here it is. It's the discussion. You've restated your results. You've explained what they mean. You've explained if they support your hypothesis or not. You talked about the flaws in your experiment. And then you say, And look, here's another way. Here's some, at least one, right? Right, at least one. Here's another way that I can test my hypothesis. And again, like I said, um, you want to use another example. So here's an example that you cannot use. So I try to come up with an off the wall one. Rather than observing the pH change in a cuvette for 45 minutes, which is what you're doing, right? Whatever experiment you're doing, it all boils down to that, right? That's what you're going to be doing. What you could do is put algae in 2 fish tanks that have different amounts of light and observe their growth for 45 days, right? So there we go. That's a way to test the hypothesis for the first question. Like, does light intensity affect photosynthesis? Well, how about, again, like, just to describe this, we take 2 tanks, both of which start with the same amount of water, the same amount of algae, the same type of algae. We let them exist, let them grow for 45 days. We measure them once a day or whatever it is you want to say, and then, to see which one grew the most, right? Which one has more algae at the end of the 45 days. So again, just an example. You can't use that one, but just, the world is your oyster. Think of another way to test your hypothesis. All right, these last two are really pretty quick and simple. Figures and tables. This is very simple. You've already done figures and tables. You do figures and tables in your lab. Literally, in the lab, you do the lab worksheet. You can copy it and paste it into your lab report. The only difference is, and actually this isn't technically a difference, because technically your table should already be labeled table 1, and then it describes what's in the table, right? So your table needs to say that, table 1, and then a brief description. A change in pH of cuvettes filled with algae at different light intensities, whatever it may be. Pretty simple stuff. Include your units. So if you have a table, and it should look like this, basically, right? Where we have a pH, right? pH for one in the dark, one in the light. Of course, there's not. Well, yeah, there's one like that, right? You'll be putting your pHs there, blah 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 blah, and you check every 5 minutes, right? So 5 minutes, 10 minutes, 20 minutes, whatever, 15 minutes, 20 minutes. Obviously, if you just put numbers there, that's not going to make sense. So you need to put somewhere like minutes, right? Time, in this case, minutes is your units, right? We're talking about 5 minutes. We're not talking about 5 hours or 5 days, whatever. So just use units. Graph, again, very simple. The only thing I really want to point out here with the graph is previously, actually, and also for the rest of the semester, you can use paper graphs. You can do a handwritten graph, but this is the one time you need to do on by using a computer, and we can help you if you want. Again, you've learned in the first lab how to make a graph. So if you need help, obviously come to us for help, but also refer back to the first graph. All the information, the first lab gives detailed instructions on how to make a graph. I recommend using a scatter plot graph, right? That shows the line for each cuvette. So say, for example, we have one, I don't know, light versus dark, right? So here on the y-axis, you have pH. Here on the x-axis, you have time. Parentheses, don't forget your units, minutes, right? And then you have the pH in the cuvette in the dark started at whatever pH that is, slowly went down maybe. And the pH in the light started here, slowly went up, right? And then you would have a key somewhere that says, solid line is light, dashed line is dark, whatever. So that's basically how it's going to go. Now, I said I recommend that, right? So I recommend a scatter plot graph. Now, if your data is such, like I said, a lot of times we can't just look at the beginning and the end of the data. We also need the in-between data. But if your results were in a situation in which you really could just look at the beginning and the end, then that's fine. In that case, you'll have a bar graph, right? So, you could say that the light, the one in the green light increased its pH by that much. The one in the red light increased its pH by that much. The one in the blue light increased its pH by that much. The one in the white light increased its pH by that much. I hope that makes sense. If not, let me know. But anyway, again, you can use either graph. But there's sometimes, you be careful if you try to use a bar graph, because again, if the data I shouldn't have drawn that yet. If the data was something like this, there we go, to where those two bars are the same, right? Let's assume that's at the same line right there. Then a bar graph isn't going to be useful, right? You might need to tell the whole story, in which case you would use a line graph. So, yep, that's it for that. And then the really, really easy section is literature cited. This should be very simple. The only thing you need to write this whole lab report is your lab, right? All the information you need is in the lab worksheet. However, if you do look out and get different sources, you just need to list them, right? So if you say something, here's a good example. If you go into detail and mention the fact that, I don't know, carbon. So, for example, you have to talk about how carbon dioxide is produced, right? That's an important thing in this discussion. So, if you go into detail and say something like carbon dioxide, instead of just saying carbon dioxide is produced in photosynthesis, which is what the lab says. If you go into detail and say something like carbon dioxide is produced, excuse me, I'm talking about respiration. Carbon dioxide is produced in the citric acid cycle of respiration. In that case, that is information that did not come from the lab itself. It came from the textbook, presumably, right? So you'll just list that in your literature cited. But again, you do have enough information that everything in your lab should be all you need. And again, if you come with information that's not in your lab, you need to include it. You will lose points for that, even if it's something that you should know because I told you. If it's not written in your lab, then you need to cite a source. And the source could be your textbook, but you better make sure it is before you cite it. The format doesn't matter, right? I don't care if you do APA, MLA. I don't care. This isn't which is kind of sad. You should be better at communicating as a college-educated citizen. But I'm just so focused on the science of it that I'm not worried about how you list it. The important thing is if you list a source that's not your textbook or your lab worksheet itself, I need a link, right? I don't care how you write it, but definitely give me a link. I need to be able to click that link and read exactly what you are citing. And then one last example for that. For example, do not put www.nationalgeographic.org. That's a big old website with a lot of stuff, right? You give me the exact link that's going to take me directly to the exact article or source that you used. And that's also great advice for independent work too, by the way. So that's it, literature cited, and we're done. The last word for attendance will be discard, as you can see right there. So make sure you get this done. Of course, at this point, you've watched the whole thing, so I know I'm preaching to the choir, but this has to be done before you can come into lab. So I guess maybe if you're watching it and you know somebody else hasn't, you should tell them, make sure you do this because you can't come into lab unless you do. Yep, that's it.